Hi, my name is Taran Solati. I'm the Associate Dean Research and Innovation in the Faculty of Forestry. It's my great pleasure to welcome all of you to our research seminar in April. Before I start, I would like to acknowledge that the UBC's Point Grey campus is located on the traditional, ancestral, and unceded territory of the Musqueam people, and it has always been a place of learning. I also recognize that those on Zoom joining us today may have been located in different places, and I acknowledge the traditional owners of those lands as well. Before I start, I would like to also thank those who helped with the seminar today. I would like to thank Frederick and Louise with the AV system arrangement, Zoom connection, and also registration. Sonia and Jillian with preparing the poster for the seminar and also advertising that. I would like to also thank Karen and Nicole for ordering the food, setting up the room for us, and bringing the sandwiches here. Now, I'm pleased to introduce our speaker today, Dr. Jaya Joshi. Dr. Joshi joined the Department of Wood Science Faculty of Forestry in 2023. Specializing in synthetic biology, her expertise lies in the metabolic engineering of microbes using tailor-made crystals. Her primary goal is to develop innovative methods for transforming biomass feedstock into valuable products, ranging from fuels to pharmaceuticals. In another word, changing trash into treasure. Her research navigates the landscape of enzyme design and evolution for radical metabolic engineering. This exploration is driven by her commitment to realizing sustainable carbon farming by 2050. So please join me in welcoming Dr. Jaya Joshi.
Thank you very much for coming. I wasn't expecting this full and then on Zoom also. So before even starting, I would say that till last week, I made it totally scientific and all, and I talked to Tarani, and Tarani is like, Jaya, make it simple. What you do, not everybody is going to understand. And that's why I have made it more like a simplest way of my research. But anytime, if you feel you have more questions or if you have like some scientific argument about our approach, please feel free to come to me and I'll be very interested in discussing that in a scientific way more. Okay. So first, I would like to do the land acknowledgement. We would like to acknowledge that we are gathered here on the traditional, ancestral, and unceded territory of the Coast Salish people, including the territories of the Musqueam, Squamish, and Watuha nations. And even some of us are coming from a different land, so I would even try to acknowledge those people where we did our research in the past. So I put this slide yesterday because a student came to me and he asked me, do you know who is Dr. Jaya Joshi? And that's where I felt that maybe a lot of people don't know me because my office is a corner office and most of the time I'm just sitting there in my silo. So I realized I have to introduce myself first. So given that I come from this beautiful place in India, which is a small community near Himalayan region, beautiful place. I moved quite a bit, almost like 11 cities. And then I went to do my postdoc in Florida. It was beautiful. I came back to Montreal and saw this. So then I realized I have to go somewhere where I get everything. And that's where I landed it here. So this is me, that I define myself as a plant biochemist. My degree was in plant biochemistry. And then later I moved to synthetic biology. And what I'm interested in is known as the enzymes that are not good enough. So I just go around and look for enzymes which are not doing great. and I try to make them better. That's how I define myself. So coming to what I do here. So my lab is known as the Biocycle Lab, and we do revolution by doing enzyme evolution. So that's how Harani defined that we do enzyme evolution, editing, and most of the time engineering, which is very different than what forestry other scientists start doing. So it's like I'm a new addition here, which is trying to do something revolutionary with the help of other scientists. So and our motto is to reduce, reuse, and recycle. So all our projects are based on these three signs, which is reduce, recreate, and recycle. So if you just think about today's world, this is what you will see, that total world population is 7.7 billion. And by 2050, we have to feed 10 billion people. So that means population is growing. And then we have other challenges like fruit production should be double to reach up to 70%, and we are wasting 30% of food. On top of that, 2050 is also a date when we have to reach zero CO2 emissions, and all by 2050. So we are running late. So what we could do, the only thing we could do is this. We have to think about how to improve photosynthesis. We have to improve the yield. We have to find sustainable solution for waste. That means we have to do carbon revolution. And that's what is the topic about today is long live carbon revolution, because that's what we have to do, and that to do faster. So what happens that when we want to think about the carbon revolution, we are thinking about 3 Fs, food, feed, and fuels, because that are the major components. And like I said, we have to do some revolution, but how we can do revolution, from my point of view, I do it by using this word, which is known as the enzyme. So we are using enzyme to do the carbon revolutions. But how I'm using the enzyme to do this, bringing this revolutionary things here. So what we use is called the synthetic biology. So feel free to think that I don't know synthetic biology, because not everybody knows synthetic biology, because it's a relatively very new field, which was emerged like 20 years back. So given that, if you ask me what are the colors of the revolution, there is a red revolution or red biotechnology, which is known for the vaccine and pharmaceutical. There is a white one, which is known for the industrial biotechnology. Then there is the green one, which is mostly known for the environmental and ocean biotechnology. And there is a green one, which is everybody knows that this is related to the crop or food related biotechnology. So when I'm talking about these ones, one thing combines them all, which is enzymes. So if you can revolutionize the enzymes, or if you can bring some revolution through enzymes, then you are going to affect all these kind of revolution, all different colors of revolution. And that's why I am targeting enzymes as a revolutionary point to target all these kind of biotechnologic eras. So I would like to ask you, how many of you can identify at least one of the picture, or think that I know this thing? I'm pretty sure everybody knows at least one. That is COVID vaccine. That is, everybody knows it. Can anyone know 2 of them? Okay, maybe Impossible Burger? Yes. Then there is third one, which is a bio-nylon. Do you know bio-nylon? This is the new product in the market. It is green, it is biodegradable. Imagine you wear your shoes, then you can throw it after 2 or 3 years. It's a biodegradable. Then there is a leather in the lab, which is actually produced from the mushrooms. And then there is a perfect day animal-free milk, which is also. What is common about all these things? These are products from synthetic biology. Every day we are consuming them, but we don't know synthetic biology, because it's very common that synthetic biology in past was defined like that. When I was in a school, I had no idea what, and this is the definition from there, when they talked about that synthetic biology is designing non-natural synthetic molecules. And everybody thought we are doing non-natural things with the synthetic biology. But now in today's world, it is defined like that. It is redesigning existing biological system to fulfill some range of application in modern day life. So it's like we are doing something engineering with the biological system. So it's kind of a combination of a biology and engineering. So how you can connect yourself to the synthetic biology? These are some things which you can read, which is in textile industry. Then you can eat all these burgers, which is like plant-based burgers are using synthetic biology a tool. Then you can have even cannabinoids. No need to grow cannabis in your backyard. Just you can come to my lab and do some yeast engineering to do that. Then you can even do that if you don't like all the perfumes which are available and you want to create your own, you can do synthetic biology with it. Even in Brazil, they are trying to run their buses using the biotechnological tools like a synbiology so that they can replace the fuel conditions there. But the thing is, it's not simple. It's not easy. And that's why if I want to bring the carbon revolution, I do want to know whether I can have better enzymes, better microbes to perform this thing, and better production. So my lab actually focuses on all three aspects. But today I'll focus on the better enzymes first, and then I'll take you to this journey. So this is what we are trying to do with the enzyme evolution. That we are learning from our evolutionary path, and then can we use exact same principle to bring the enzyme revolution in today's context. So I added this slide yesterday because I realized a lot of you might not even know that what is the central dogma of life, which is very simple. If I say you have a DNA, it goes to RNA, you produce some amino acid goes to protein. Then what we didn't learn that protein end up in the metabolites in our body. And these exact proteins are also, and enzymes are these proteins. So if I change anything in my amino acid, I'm gonna be changing my enzymes. And that's what it is. And if I'm changing my enzymes, I'm changing my metabolite. And to change my metabolite, I have to go back and do DNA change. So actually everything is connected. So evolution is actually like some changes happening in your DNA, which is affecting your enzymes. Enzymes are affecting what you produce, and that's how the evolutionary process works. So we take advantage of our oldest goat. Half of you may know him. He is Darwin. And Darwin was known for giving the theory that there is going to be a slight variation, and if they are useful, by natural selection, and that will create something new or something better. And what happens with the evolution? That it creates variation. That's why we all are different. Some people are better than others, while some people are better in doing coding, some people are better in doing arts. That's how you can see that variation always helps you. Then some people have a fitness difference. Some people are athletic, some are not. So that's how the variation helps you. The mutation has created in us, and then it's heritable. That's why a lot of our siblings are similar to us. That's why, because we got everything herited from our parents. So that's how the heritage. So whatever mutation you are creating, that helps you to create these three things. And in my lab, we take advantage of this and we learn enzyme design space. I'm going into a little bit detail here, which is going to be a little bit tricky, but feel free to ask me any question here. So enzyme design space is actually a mathematical concept. So if I come here and say that it's vast space, you can make n amount of combinations. You know why? Because there are 20 amino acids which are existing in our body, known 20 amino acids. So if I make a combination of 20 amino acids for a 1 kilo base pair long DNA, you can see this calculation. I'm just showing you there. 300 residue means 1 kilo base pair long DNA has a 300 residue, and this many possible combinations are possible. If you want me to put it in a context, there are 10 to the power 80 atoms in universe. So it's more than that combination. But technically, it is not possible. There are only very few possible enzymes and pathway exist. It's not that all sorts of combination are there and I have to just pick it. No. And that's why, if you want me the context in life, if I want to change glucose to pyruvate, which is a very common, everybody is having that pathway in their body, we have just two ways to do it. But if I calculate it synthetically, the numbers are like this. There are 12,000. So what is happening actually, this is originally what is happening with us. And if you come to my lab, I'll be doing something like this. So we will be doing all sorts of combination, but that's how the enzyme design space is very complicated to understand. But then I'll take you back to my landscape to clarify how it works. So you have an initial activity of an enzyme, which is a wild type, which is not good. You are like, okay, it's working. You want to take it to the top. So either you will take an easy route and just say like, okay, I'm better than what I was. And that's what we call it a local maxima. That means you did few changes in your DNA, then enzyme, and you are happy with it. Some people are very adventurous. They want to go to the valley and then they go to the top. That's where the complicated stuff happens. But this is a very tough task to achieve. And there are a third kind of people. They do this. They go there, take a break, and then go up. That's how you create mutation. You are like, okay, I made it better. Maybe my Rex down, I'll make it much, much better. And then there are a few more. They're like, I don't want to be that better or something like a maximum one. I'll just go to the different peak or do some different kind of activity with my mutation. So that's why enzyme design space is a very complicated stuff. And that's why you should be having a very good system to do this kind of enzyme evolution in your lab. Because outside nature is doing it, but you want to do it in your lab. And that's why my lab uses microbes as a system where we can do this job. Okay, so what happened that I did my PhD, and that time my enzymes were very bad. Till that time, I had no idea, 2018. And I wanted to make my enzyme super catalyst. And you can imagine that was so bad enzyme that I was like, oh, I'll make it super catalyst. But it never happened. But that time, 2018, Dr. Francis Arnold got the Nobel Prize for doing directed evolution of enzyme in lab. And I got interested in this whole thing because I was like, oh, I have enzyme and I want to make it better. I didn't know that she took like almost 30 years to do this. And I was imagining I'll do it in the next 2 years or 3 years. So I started doing my PhD with this. But first, I wanted to know that how the mutation works. So I just put it in a context for you. You want to mutate one of the enzyme, which has one amino acid. You will mutate, you will choose, you will mutate, you will choose, you will mutate, and you choose. And that means it will take me ages. There are some mutation experiments running for 40 years right now in the labs, which are doing this, just to see how evolution works. But what I'm using right now is called in vivo system. I'm not doing it all by myself. I'm not hiring anyone to do it. I'm hiring microbes to do it for me because they divide very quickly, so they can mutate and do it quickly. And that's how I ended up. I am thinking of that I'll make super catalyst in next future. That's my idea. So to just put you in context in the last one, that I have an enzyme which is A, I will take it to B, and then I'm also thinking to take it to C. And what I'm going to be doing, I will be exploring all sequences space. That means all combinations. But I'll be putting some kind of fitness test there. That means putting some pressure from outside. It could be like acid test, or it could be like a growth test. And whatever survives will go up. Exactly what Darwin said. Natural selection will take the lead here. Okay. So in my lab, we have 2 enzyme evolution platform. Which is in yeast. So in yeast, what I'm doing here, you can see the target gene. So I'll put my favorite enzyme here as a DNA. And then I have a faulty polymerase. In all of us, there is a polymerase which works on DNA to replicate it. But what I'm putting here, I'm putting a faulty or an error-prone polymerase, which will create mutation. And that's how I'm using yeast as a mutation agent here. Another system which I'm using is an E. coli system, which has the exact same theory. But what will happen, I think a lot of people asked me earlier that, Jaya, plant enzymes, you can't evolve in E. coli because this is bacteria and plants are very different, prokaryote versus eukaryote. So I'll do it based on what kind of enzyme I have. If I have a plant enzyme, I'll go with the yeast. If I have a bacterial enzyme, I'll not go with the yeast because yeast is already complicated. So I'll just go with the E. coli system and work with it. So that's how we are using these two platforms in our lab. Okay, and it's a stable, it's scalable. You can do it like so many times and it's very useful. Okay, so now I have evolved enzyme. What could I do? Whatever you want me to do, which includes I can improve Rubisco, which is a great deal. People are working on it because they want to improve photosynthesis, improve yield. It is like a very common enzyme. You can work on the food and feed. They made impossible burger. We can make our own sandwiches here, which will be more tasty and whatnot. You can make the bioplastic and biomaterial. You can add functionality to your clothes by using these enzymes, right? They can be like, oh, I want them to have like some UV reflection or something, or even lights. People are trying even cathode anode kind of enzymes, which you can add to the. Then you can add the biochemical and additives, and ultimately you can work on the pharmaceutical and vaccine industry also. I'm not doing all of these. I have picked few to just showcase you, but the future is like enormous. You can apply this enzyme engineering technology to any field right here. Okay. So in my lab, we have selected few enzymes right now, and we are trying to evolve them, and then they will be used in different. I have used plant enzymes. I have used yeast and bacteria to see how it works in Vancouver weather. Because whatever I have developed, these technology were developed in US, I bring it with me. But again, there is gonna be a long path to reestablish it here and do it. So we are just testing few enzymes right now, okay? So now I developed the enzymes. What we are doing quickly, I'm putting it into my motto, reduce carbon loss. So you all are from forestry. And if you have 100 photosynthesis combined, how much you know that you can get it as a yield? How many carbons of 100 of photosynthesis you consume goes into yield? How much percentage I should ask? No idea. So if you fix 100, you're not forestry. So if you have 100 carbon fixed by photosynthesis, 50% plant uses for themselves as a respiration. They just give you 25% as a harvestable yield. 25% goes as an unharvestable yield. And then it's like that. If I divide even respiration, one plant uses for their respiration, one for to synthesize their enzymes or something, whatnot, okay, for their own purpose, like for their own body. So that's why I am just showing you that you can do two things. You can increase photosynthesis. That means you order bigger pizza to feed more people. That means you improve photosynthesis Rubisco, or you can reduce the maintenance cost. So you will actually affect the, size of the harvestability. You'll make it big, people will harvest more. So what you can do next is whatever extra biomass you are doing with this process, which is enzyme evolution, you can use it for many purposes. In US, people are trying to actually capture more carbon with this method and trying to bury it underground so that you can cut the carbon from the environment. And second, you can use that carbon for any purpose in later years. So that's how people are doing it. And our plan is to design more pathways there, designer pathways, and make more biopolymers kind of thing. So that's how we are thinking that that's how we can reduce carbon loss here by using this. Okay, so we are doing redesigning. So one example was that, which I used in my postdoc, that there was an enzyme which was very bad. That means it used to do one cycle and dead. after one cycle. So what we tried to do, that we tried to calculate that how much maintenance cost it is for a plant to resynthesize this enzyme again and again, and it was very high. So what we did, we tried to do the enzyme engineering on that enzyme, and when we calculated, we were able to improve the yield gain from 1 to 4%, which is a huge in crop breeding aspect. So that's why enzyme engineering is important. You have to think through where you want to put it in. Okay? So what we are using in our lab, which is enzyme evolution for green biotechnology. So like I explained you before, that there are different colors of biotechnology. For green, it is mostly used in agriculture or agriculture related products. Some people are nowadays, you will hear that they are using algae to produce a lot of things like cyanobacteria, because these are like a green plant-like structure, which can give you a lot of food feed related prospects. So our lab actually are thinking that how we can contribute for fuels, for food or feed and trades. So that's how we are thinking, that how we can make surplus carbon and then use it for these kind of things. The second one comes that we are trying to recreate cell factories. So I said, we can do enzyme, we can make them better, but you can't keep those better enzyme on your shelf here. You have to do something out of it. So what you can do, you can create green factories. That means those microbes, you can put it in your, so what you do for the bread making at home, you use the yeast to make the bread for you. So you are actually running a bread factory in your kitchen. And that's what we are trying to do. We are trying to create these cell factories in our lab. And that's one thing, which is, that's how we are contributing towards white biotechnology. So white biotechnology is using microbes to produce a lot of other stuff, which could be fuel, which could be cosmetics, which could be textiles or plastic or whatnot. In terms of cosmetic, right now, my previous lab was working with a company, and they have now created some of the bio ingredient, which used to be from shark. We used to get it from shark, and it was added in our all like moisturizer and all whatnot, like good looking creams or not. But now we are actually producing in our lab, so that we can use that ingredient, and that means you are also not affecting shark and their population and biodiversity. So that's how synthetic biology is playing big role in your day-to-day life or your future, for like whatever you are designing for 2050 or for your kids. So coming to this, so how we can do it? We are taking bunch of enzymes. Some designer, some are not designer. Some are like, good, I can use it from plant. I'm putting in yeast. Some I'm designing it for the yeast. And then we are putting everything like this. We were taking to the whole like maxima, local maxima and best ones. And then we are putting in the yeast. So that's how we are thinking that we can create some biofactories which will be producing something useful. Okay. Now I have another question for you. I have put these fruit pictures here. From your childhood, can you tell me what your parents told you? What's similar between the kiwi and all sorts of oranges? What's the key ingredient here? Yes, Felix. Vitamin C, yes. So why I'm putting the vitamin C here? Because I'm working with vitamin C. The problem is right now, whatever vitamin C you are getting, because nowadays nobody eats this side, eat the middle one. So whatever we are getting is actually produced from the petroleum-based process. That means it's very expensive. People are using it, and it's using a lot of energy, and whole process is very expensive. So what I'm trying to do is I went back, decoded the literature. So it looks like plant and animal produce these ascorbic acid, and that I'm like, why not? I can put it in the yeast and let yeast produce it. Okay, so that means you can see in my lab, you can get the beer, you can get the baker's yeast, and then maybe someday you will get some ascorbic acid tablets in my lab. But there is a twist here, that I'm putting this pathway in the yeast. But you know what? I'm using waste to produce these. Any food waste, feed waste, or any forest waste will be used to do this process. That's why we are introducing a full pathway which will use the forest bioproducts or any food waste, and that's how we will be creating ascorbic acid. So that means we are actually contributing for the next aspect of our lab, which is recycle feedstock waste. So that's why, like I said, our all projects are running on this motto, how to use these reduce, recreate, and recycle. I like this quote a lot. Then my mom used to tell me that waste isn't waste until we waste it. So this is so true in my lab that we don't want to waste anything because it helps you to feedstock valorization. That means you can do something useful from it. And then there is a circular bioeconomy, which we talk a lot about this now. So that's why Tarane said that I try to take whatever you are throwing in the sink to shelves or whatever you are throwing as a trash, I'll use it to make some treasure out of it. So sink to shelves, this is one story which I'm still working with the Concordia University, the AgroPro Dairy Cooperative, whatever milk or dairy products you are taking nowadays, you will see this logo. This is the biggest cooperative of Canada. And they came to us and they said, hey, we have a lot of waste, which is known as the whey or the milk permeate. It is a waste and how we can use it. So what we said that, okay, we will take a yeast, we will put a pathway in it, which will utilize your cheese waste, and then we will convert it to fumaric acid. And this fumaric acid, you can sell it on the shelves, which will actually is a food additive, which is actually in US, it has on a high demand right now, food additives. So that means whatever they were throwing in the sink, we were planning to do it for the shelves. Okay, so this is one example we did in my lab. Because I'm in wood science, I'm in forestry, and I feel all about the feedstock waste. So this is a very well-known thing that if you do a digestion and pre-treatment, you will have a lignocellulosic biomass, which will be rich in sugar and phenolic acid. So a lot of time people think what we could do with this. People come up and saying you can do a lot of things with it. And how you can do a lot of things? By using microbes as your cell factory. So that's what we are planning to do, that how we can use this waste and use our designer enzymes to do something creative. And from this, so a team here with other researchers from our department, we put together this a project, which is a forest produce in a Petri dish. That means the plan was to look outside and look for the waste, and can we produce bacterial cellulose from it? Bacterial cellulose is very much like a cellulose, which is plant, but it is more purified or have a very good tensile strength, and it is now in the market and people are trying to produce this more and more. So what we did that we have these bacterial strains in our lab and we are trying to engineer them so that they can eat our feedstock, which is rich in xylose and other sugars. And that means we will use the forest byproduct to produce bacterial cellulose that could be used in the textile industry or medical industry and packaging industry and whatnot. So the plan is very simple, looks like simple. It's not simple. That means we have a feedstock waste. So in your lab, if you are generating any kind of feedstock waste, please contact me. I'll take it. Because we are looking for a different feedstock waste, which we can use to do some kind of a bacterial cellulose production in our lab. The idea is to have a high value bioproducts. That means we should produce this good. And I think with the Dr. Ching Shi too, we are also doing LCA analysis, whether it is economical to produce, or TEA analysis, whether economical or not, because everybody is producing bacterial cellulose. But when we are thinking from the waste, it's a unique idea, but at the same time, is it economical and whether we will be able to produce it enough or not? So this is one project in my lab. Yeah. So this is all about that three principle I told. But the thing is, whatever I do, I can't do it in silo because I do enzyme engineering, but to put it in the bigger production and all, I always need two things. One, funding agencies. So in past, I was very lucky that I had money when I was working as a postdoc and all, but now we are looking for a different funding agency who can help us to actually give us some kind of funding to run these proof of concept experiments in our lab, because it will take another like few years to get it done. But I was also lucky to have like three amazingly great plant biochemists and synthetic biologists as my mentor. And with them, I was able to develop a pretty good one. I would like to continue the same thing here. And that's why when Tarani asked me to do this, I was like, oh, this is a good that people should know what I'm doing so that tomorrow they can come and have some collaboration with me. Okay, so that's why I'm looking to have that collaboration and diversification kind of theory in my lab, and which is important. So till now, I told you there are three pillars in my lab, which is reduce, recreate, and recycle. So what's the foundation of these pillars? Actually, equity, diversity, and inclusion is the one foundation in my lab. I work in a very different kind of field, which will have not many people know about it, or many people are not have any experience in biology. So my idea is to bring whosoever is interested in these kind of ideas, maybe from plant biochemistry or even a forest byproducts side of it, or even microbiologists. Whosoever has some idea of it, I would love to have take them. My lab also runs on some of the machine learning kind of thing. And the idea behind that is to educate more and more women on this part of the science, which we have seen a very few. So if any one of you are interested in running those workshops where we could help women or even like a student to learn coding as a workshop, I would be very happy to organize those workshops as like a foundation of my research. So with that, I would say that I was very fortunate to work with this different groups here and in US. But right now, and I was lucky also with the Canadian government that they gave me some funding to run the projects when I was in Canada. But right now, you will see some of these faces. Even I don't have some pictures here, but these faces are newcomers. They are joining my lab. They are new to UBC. They are new to forestry. So I'm showing you these pictures. So if any one of you see them lost, just like me, just feel free to help them. That's why I'm showing you these faces. They are gonna be joining from May, and they will be helping me to understand. But if any one of you are interested in running the SynBio pilot projects or learning about SynBio, I'll be very happy to get that collaboration going. So with this, I would say that, like in the first slide, I was saying that carbon farming, everybody has to do by 2050. But from SynBiology, we are thinking we have to think through now, because it's already too late. 25 years, nothing is going to change if you don't do it fast. And that's how SynBiology is one of the solutions which you can use. So I posted this slide when I did my interview, and I again say this, that you don't want to leave your child or your children with this environment where everybody is just thinking about how to capture carbon or what to do it and how to reduce waste and all. You have to find the solution. And synthetic biology is one solution which will help you. So that's why synthetic biology is revolution, enzyme is revolution. Enzyme evolution can bring the revolution, and that's how my lab is doing this. With this, I'll conclude my talk, but I feel free to ask me any question, science or anything outside of it. Thank you so much.
Thank you, Jaya. That was excellent. I really learned a lot. Any questions for Jaya? Yes.
Thanks, Jai. That was a very, very interesting talk. Lots of questions, and maybe we should have a chat outside at some point in time. But I did have one question about the feasible space that you showed. You showed basically a solution space that had two peaks, one larger than the other, and there was also a trough. To what degree can you map out that space prior beforehand versus using your lab experiments to map out that space? Is it a theoretical space that you could actually map out, or and if it's the latter, that you're sort of exploring that space experimentally, how do you know when you've hit a trough that that's not just on the, and then stop, if that's not on the way to a bigger peak?
That's an excellent question. That's an excellent question. And I think that's the reason I started the machine learning platform in my lab. It's always tricky to figure it out that the student who is not performing well, whether they are gonna like just say like, okay, I don't want to do it, or they are preparing for it and go like take a big leap. So that's exactly happens in the enzyme design space, that sometimes some combinations will not work. But the moment they have one extra kind of mutation there, that combination will work, and then you will get the maximus peak. So that's why we run so that's how the in vivo continuous directed evolution experiments are helping right now, that you keep on monitoring the growth. The one who are dead because they hit the valley, but the one who are like had like that extra, that population will survive a little bit on your next round of mutation, and then you will see the peak. Without experimental thing, it's very hard to do it. But right now we have a machine learning platform which we are running, which has the data that what will happen that if you have these two kind of a combination, will you survive, or you will have a better activity or a bad one? And that's what we are mapping it out. But without doing experimental work, it's always hard to identify that whether the thing which is going towards the valley is going to be dead or not. So it's kind of a survival of the fittest. Again, we have to go back to the natural selection and see that with that selective pressure, anything survived during that valley time also. So it's hard because it's a mathematical concept. And again, it has to happen in the living system. But with the metabolic modeling and with some of the machine learning platform, we are trying to at least assume what will work or not. Yeah, but a very good question. Yeah. Oh.
I have a question regarding the mutations you do in the enzyme. So you do it randomly? You just use like an polymerase that is error prone, or have you used like, for example, CRISPR-Cas to create like more target mutations once you know something is working? And yeah, see if you can give
This
us
is
more
another
detail.
amazing question. So what happens that in past, people thought that if I mutate 1 or 2, which is known as the error-prone polymerase enzyme, a PCR. So what people did, that they put in a PCR, not a good polymerase, bad one. It did a kind of a site-directed mutagenesis. Michael Smith laboratory, which are named after Michael Smith, was famous for doing those site-directed mutagenesis. That means you are doing that mutation on the particular site and you get the better enzymes. But what happened, exactly the previous question, if we don't know what mutations are going to work, or if we don't know what combinations are going to work, so people started thinking, let's use the error-prone polymerase. So that means the error-prone polymerases, what we use right now, are randomly creating mutation. It's a very random method. It's kind of a game of Plinko. You go from like this, whatever is good, I'm good.
You have like a library.
Yes, you create the library. Yeah. So you, so because I say this, I'm lazy person. I don't want to do that library work. So I give it to yeast and it does the library for me. It is like, I'm just replicating it continuously, and whatever survives, I'm thinking, oh, this is good. This might not be good, but I have the idea that it is surviving, so it might be doing good. But the third one, which you said, the CRISPR one, there is one system known as the Evolver-R, which uses actually a Cas9 enzyme, which actually does more kind of a mutation. But the problem with that is you have an enzyme and Cas will just target few areas, like 60 base pair 60. So the efficiency is not that much. The error-prone polymerase actually takes full gene and do boom, 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 boom. And it is like you have a lot of combinations. So there is a random mutation, and then there is a site-directed, means you know which site I want to do. And then there is a combination of both. You do which you know will work, then you know some, you want to do random. and some you want to do with the CRISPR editing. Yeah.
Great talk. I want to ask you a quick question about your strategy. So when you're doing enzyme evolution, especially when you're looking to make a product, most of the time it's a sequence of 6 or 7 enzymes. And so as others have probably thought about it, you may not have the bottleneck. You may not have the enzyme that's actually the limiting factor. So why would you choose this approach over not just trying to do something like genome shuffling? Just let nature, as you said, do it itself and then pick that organism and figure out which genes and which proteins were modified to make it a better organism.
I think that that I come from a lab which was doing in genome shuffling. And what I learned from the lab, and even now we realize that the genome shuffling is a kind of a tedious process. You have to have your hands free to do those genome shuffling experiments. And that also one more, which we actually came across with the genome shuffling, we didn't have a haploid population. So if you have already an organism which is diploid and you have to make them haploid, they should be stable haploid, you have to mate and do a whole genome shuffling. So that's why genome shuffling is a wonderful approach when you are just using SAC as the one, because a lot of, but when you are gonna go in the territory where there is a lot of unknown about genome and all. So I think combination of directed evolution of enzyme and using some enzymes from outside, and you can install those enzymes, will work. But I'm not against genome shuffling. It's just that I felt that it's more tedious process, and why not I use a in vivo continuous directed evolution for that. But genome shuffling was successful in past. A lot of people were very successful, but I also understood that it's a tedious process, and having maybe you might not know, but UBC, one of my reasons to come to UBC was another, that we have here like a biofoundry, which has robots to do it for you. So I'm like, why not go there and then have those kind of experiment, which will have making a different kind of libraries, and it will be more targeted also. I feel that you are not going to be doing much, but yeah. So once my previous boss retires, I will take genome shuffling projects also. Yeah.
Thank you for that very interesting topic. You choose for bakeries. So question for you is, so the waste that we're using, everybody is treating with the waste. You see to use the organic waste for energy, biomass energies. I'm not sure if you consider about the energy engaged in your enzyme development. Is this sustainable, or comparing to the conventional way, the other ways? How great you are in terms of carbon reductions?
Yeah, no, I think what I was trying to show here, I think biofuels is like literally one area where enzyme evolution is gonna be playing a big role. Technically, biofuel is not my area. I have not worked around it, but I have known that in US there are like big institutes which are actually working on using the enzyme evolution techniques to work on the biofuel side of it. Sometimes they even improve the yield in the crops, which are actually good for biofuels. That's one way. And sometimes with the waste reduction. I was reading one article somewhere, and they said that, oh, now the waste will run the aircrafts. And I think the whole theory was that you can use the waste to get the biofuel side of it. But I have my personal thinking, it's not going to happen in next future soon. So you are not going to be running. Brazil is not even running yet, the buses, which they thought that 10 years back. So it's going to be a place where people do need to put more efforts. Yeah.
Okay, thank you. Thank you for the presentation. Now I have a much better idea why we hired you in this department. One thing that we forgot to give us when you went through slide 3 is the room number of your lab.
Oh, okay.
In the beginning of your, yes, there are some interesting things in there that we might want to use. I have a more philosophical question about you. Long time ago, when genetic modification and gene manipulation started with food stuff, everybody was excited about the end of starvation. GMOs were the answer of increasing food production for an increasing population. Fast forward today, nobody wants to consume GMO food. Okay, so synthetic biology also is an exciting new stream of things happening with, manipulating natural stuff, let's call it stuff, organisms. Do you think like 20, 30 years, people might have the same attitude towards synthetic biology? Yes, no, and why?
First thing, I was part of the genetic engineering era. I worked for Monsanto, which was very infamous to do all that thing. So I was part of it. I felt one thing where we were doing, it's more ethical issues, which came with the genetic engineering, especially with the food. I remember my parents telling me that don't do anything with my food. I'm okay to take any pharmaceutical or any vaccine or anything, but don't do anything with my food. And I realized this is more about the food. So even when I'm talking about bringing the synthetic biology in the plant world, I'm not saying I'm going to introduce a foreign gene in plants and say, tell my parents consume it. What I'm doing, actually what she said, that using the CRISPR, which is an editing technology, that when I know from my experiment that enzyme needs A, B, C changes, I will use the CRISPR, which is an editing technology, edit those one, and then can I do much better for my plant? So I'm not introducing any foreign thing. That's one aspect of synthetic biology. Mostly, I like synthetic biology in terms of like not just doing a genetic manipulation, but it's kind of a providing you some sustainable solution. So one biggest example of SynBio came is called artemisinin. And maybe a lot of you don't know, but I come from India, so a lot of people are affected with malaria. And a plant which has produced this medicine is called artemisinin, which was in China. And people were like, okay, let's grow, like, even if you grow like artemisinin plants in those China region, full China, you will not able to treat Indian and African population. You need so much to produce to get that medicine. What SynBio did at that time, they used the yeast to put that plant pathway in it, and they produce artemisinin from it, which is like a very viable solution. And that means people are using it, and then you were able to save. And exactly it's going to happen with the vaccine. That then you can't produce it in a large quantity. You might be using some other microbes or some system. So personally, I feel that synthetic biology provides solution for the future, which is not related to just food or affecting your food, which is we all are very concerned about. The SynBio is like doing a little bit further than the just genetic engineering of plant or and whatever, like feed everybody kind of thing. It's not going to happen, but it will improve things. I personally feel. Yeah. Thank you.
Any final questions for Jaya? There are no questions online on Zoom. So thank you everyone for joining us. Thank you.
Thank you. Thank you. Thank you.